All right, I think that's enough time to let people trickle in and everything. So first of all, I wanted to, thank you all of you for being here with us this evening. We are very excited to have one of our student virtual lectures here for one of our newest PhD students here at Nova Southeastern University. My name is Brett McAllister. I'm the Director for Graduate Admissions for the Halmos College of Arts and Sciences, where the marine sciences, happily, thanks for me, do reside. So with me today is one of my assistant directors, Ms. Farah Amro. Thank you for being here as well, Farah. And the moment of the hour, of course, is Miss Mya Daniels. So Mya, thank you very much for being here. I know you're a very busy schedule with everything you got going on. So really do appreciate you taking some time to speak to myself. I'm excited for the lecture. And I know some of our other students as well have expressed interest about listening to you as well. And for those of you that are not watching this evening live with us, that will be looking forward to the recording as well. We are recording this, and we will, it might take us a couple days to cut it up, make sure it's nice and neat. We will send it out to all of you out there looking to watch it later. So I'm going to introduce Miss Daniels with us as well. Mya is a first-year PhD student at NSU. She earned her master's in marine science also at NSU, and is looking to pursue a career in veterinary medicine at the Wildlife Zoo Vet. She's interested in avian physiology, environmental containment, toxicology, and conservation. When she isn't conducting research on her dissertation, she works as an assistant lab staff manager at NSU's main campus and a veterinary technician at local specialty clinic. Her hobbies include weightlifting and trail running, and she enjoys spending time with her dog Milo and several adopted birds. Now, I'm going to try and pronounce everything in the title of this lecture. I do apologize. I am not a scientist by nature, so I'll do my best, Mya, so I don't embarrass you too bad. Mya is assessing heavy metal partitioning in the vulnerable Humboldt penguin. And with that being said, Mya, I'm going to turn it on over to you. If you want to share your screen with your PDF and with your PowerPoint, you can take it from there. So Mya, thank you so much for being here. Really looking forward to hearing about your lecture today. Thank you so much once again.
Thank you so much for the introduction. I appreciate it. Also, phenomenal job on the title pronunciation. I understand I have a knack for writing very long titles. So give me 1 second. Let me share my screen real quick and we can get this presentation started. All right, can you guys see this? Okay, wonderful. So thank you again for the introduction. I'm very excited to be speaking this evening with you all. Like Brett mentioned, my talk this evening is titled Physiologic Pathways of Metal and Metalloid Elimination in a Humboldt Penguin, or Spheniscus humboldti. This is the species that I'm studying for my doctoral dissertation here at NSU. I'm very excited to talk to you all about this research. So a little bit about me. I started my studies in Dr. Hiron's lab here at Nova in my sophomore year. So I actually completed my bachelor's at Nova as well as my master's, and I'm currently a doctoral student here. But back when I first started, it was not on penguins. It was actually working on a project that was led by a graduate student who became a really good friend of mine, Mickey Edwards. He was looking at the effects of El Nino on the trophic dynamics of two different species of South American pinnipeds, which are the South American sea lion and the Peruvian fur seal. And he was utilizing stable isotopes, specifically carbon and nitrogen isotopes in vibrissae or whiskers, to kind of track to see how their diets were being affected by different oceanographic processes and food availability. So worked on that project for a couple of years. And then in my senior year of my undergraduate studies here, Dr. Hirons asked if I had any interest in birds, which I did. Like Brett mentioned, I have a couple birds of my own. So I have a lot of interest when it comes to anything avian. And that's how I was introduced to this species, the Humboldt penguin. I realize I should be using my animations here. So that's how I started working on the Humboldt penguin. My project began on some archive samples that we had that were collected from a field site back in 2008 and 2009. And I was looking at trophic dynamics and sea surface temperature changes through evaluations of stable isotopes from eggshell carbonate. So the eggshell is made up of a lot of inorganic material, which is predominantly carbonate. And I was looking at oxygen as well as some other stable isotopes to see how sea surface temperatures were fluctuating over time and whether or not there were any corresponding changes in trophic level for those birds. Now, while I do personally love a good food web and I love trophic dynamics, My interests lie more so in the realm of environmental toxicants and contaminants, which informed my master's thesis and my current doctoral dissertation project. And as a product of my master's work, I did have a recent publication which came out earlier this year. Actually, it came out on Friday, but officially on their website. So very excited about that. So that's a little bit about me. I wanted to kind of provide some project justification. A lot of this talk tonight is going to be pertaining to my work as a master's student, but also the continuation of it into my current doctoral dissertation. So for this research, I looked at heavy metal profiles in eggs from two different populations of Humboldt penguins. First, I analyzed eggshells from wild penguins that are inhabiting the Punta San Juan Marine Reserve in Peru, which are pictured here on the left. And in the second part of the project, I looked at whole eggs which were collected from managed care penguins that are housed at the Burkefield Zoo, which are pictured there on the right with some wonderful keepers. Now we're going to see this terminology, the wild versus managed care, throughout. So I want to keep these in mind, where wild is referring specifically to the birds that are inhabiting that rookery down in Peru. And our managed care birds are those captive individuals. We like to say managed care instead of captive when it comes to birds that are part, or any animals really, that are part of a zoological collection. So those two terms are going to distinguish those isolated populations. Now, you might be wondering, why is this research important in the grand scheme of things, right? So investigating the heavy metal content of bird eggs seems like a niche topic, but it's really important because of the implications that environmental contamination has on the species' population health. Now, as a follow-up, you might say, why is population health a concern? Because they are in decline, not just for the Humboldt penguins, though. We're currently experiencing massive declines in biodiversity worldwide. In 2020, the World Wildlife Fund reported that there's actually been a 52% decline in global wildlife populations in the last 50 years alone. And if we look at biodiversity lost by region, we can see that Latin America is actually getting hit the hardest, with a 94% reduction in wildlife populations within that same time frame of only 50 years. Now, if we're talking about birds specifically, according to the International Union of Conservation of Nature, or IUCN, 49% of bird species are currently in decline, and that is over 5,000 species, of which the Humboldt penguin is certainly one. Of the areas that are the most afflicted by avian population loss, Peru is actually number 4, sadly. Like I mentioned a few moments ago, the Humboldt penguin is native to Peru, so it is one of those species that's endemic to the area and is being impacted by a lot of different anthropogenic or human-derived effects. While we obviously want to keep an eye on our at-risk species so that we can work to improve their numbers because we want to keep them around for a long time to come, like the Humboldt penguin, it's also important for us to be able to evaluate their populations because birds can act as incredible indicators or sentinels of how an environment is doing as a whole. That especially applies to seabirds. They're found worldwide, so they are ubiquitous, is what we would call them, and they typically occupy some of the upper levels in the trophic or food chain, which allows them to act as excellent representatives for some really critical marine ecosystems. How do we access the data that these birds capture? There are a lot of different options depending on what kind of, study design you're interested in or what you want to come up with, but it can range from utilizing tracking data if we have transponders on the individuals, tissue sampling, fecal content analysis, a lot of different options, but the technique that I chose to use was analysis of eggs. And I chose this because eggs kind of act as a time capsule where they store an incredible wealth of information that can be used to generate a snapshot of health, specifically for the mother, so maternal health, at the time of egg formation, while also allowing us to look into the future a bit and project how the resultant offspring might fare. Because what's contained within that egg shows what was present in the mom at the time of egg production, but also what's going into that chick that is eventually going to be formed. So that provides us with a lot of information about the health of the species and their environment. And because Humboldt penguins reside in an epicenter of biodiversity loss since they reside in Peru, they were the perfect species for this project. So as I mentioned before, Humboldt penguin populations are currently in decline, and this trend is largely attributable to anthropogenic or human-mediated impacts. There are 3 main threats to Humboldt penguins. These include commercial overfishing. Historically, Peru has had a tremendous population loss when it comes to seabirds. Their seabird numbers actually plummeted from 20 million birds in the early 1960s to under 400,000 birds in the late 1990s because of commercial fishery pressures, which basically just wiped out stocks of Peruvian anchoveta as well as other species, but it's really the anchoveta or the anchovy, which is a massive forage fish. It sustains a lot of the biodiversity that we see in Peru, but it's also used for a lot of human purposes and human consumption. The second is guano harvesting. So as guano was extensively mined and exported to Europe and North America for agricultural use, it's a phenomenal fertilizer. That removed a lot of really important nesting grounds, because for penguins, as well as a lot of the other endemic seabirds that are found in Peru, they actually utilize guano accumulations or guano mounds as nesting surfaces. So this further contributed to population declines that were already exacerbated because a lot of these birds were actually being harvested as food for the workers who were living at these sites harvesting guano. And lastly, certainly not least, is mining. I'm actually focusing on mining here because the environmental contamination that is caused by mining activity poses an immediate threat to Humboldt penguins, since many of the elements that are contained in the mine effluent, so what's being kind of kicked out of those mines, is categorized as heavy metals, which are a significant health concern for all species, whether it's people, dogs, cats, birds. It's a major concern. So I've got this nice little schematic, little sunset scene here that we can imagine in Peru. So Peru is actually the second largest producer of copper, zinc, and silver in the world, and South America's leading contributor of gold and lead, the third largest source of tin, and the fourth largest source of molybdenum. Kind of a rare metal, but it definitely comes into play when we talk about heavy metal toxicosis. Ore production occurs across approximately 200 mines in Peru, and the country's largest open iron ore mine uses 4 million tons of ore annually, and it is within 20 kilometers of the Punta San Juan rookery. So I've got a little diagram here. We've got our mining site, and we have our rookery where our birds are. Pretty close proximity. We also have some expanding copper mines that are really close to the study site as well. There is an expansion that's located in Tacna, Peru, and that manages the world's 5th largest copper mine, as well as silver, gold, and zinc mines. And contaminants from these mining facilities can be brought to the coastline through rivers or any other kind of water body that is discharging into the marine environment, right? So our coastal areas. Specifically, there are a lot of rivers that are passing through the Marcona district, which is where Punta San Juan, the study site, is. So there's a lot of runoff potential from the waste retention ponds where flocculated debris is actually consolidated before removal and transport for disposal. So it's condensed into a single area. And unfortunately, if you have a lot of rainfall in that region, it'll then wash off and into your coastal marine environment. Now, this runoff is entering the marine environment and being taken up by different organisms. So we can see we have our birds, we have our fish in the diagram, and these organisms that are implicated can include plants, invertebrates, fish, the whole gamut. As the metals are acquired by lower level trophic organisms, so it might be our invertebrates or our small forage fishes, and they are transported through the food chain, we see an effect that is called biomagnification, which is just that contaminants are being increasingly enriched into the organism's tissues as we look at animals that are occupying higher trophic levels. So when we get to those big fish, those birds, sea lions, anything else that might be present in the environment. This phenomenon occurs in tandem with bioaccumulation, which is just the retention and accumulation of contaminants within an organism over the course of its life. And the concern with the elements that are present in mine efflux, so if we have, for instance, mercury and lead, is that it may constitute what we consider to be heavy metals, which exhibit some toxic effects. So in this image here, we've got some organs, right? We've got the liver on the right, we've got the kidneys up top, and then we have, kind of hard to interpret if you are not super familiar with physiology or anatomy, but it's some adipose tissue, which is essentially just fat. When we see contaminant accumulation in some of our higher level species, like the Humboldt penguin, we start seeing a lot of negative impacts, particularly in the organ systems that are responsible for clearing toxins, like the liver and kidney, or for some that exhibit physiologic proclivities for metal accumulation, like fat or adipose tissue. There are certain mechanisms that basically allow for a lot of metal retention in certain organs that are either constantly kicking it out, and occasionally you just have some arsenic or some, molybdenum or lead that's going to stay in those tissues, doesn't actually get eliminated properly. Or in other cases, when it comes to lipids, there's a lot of interactions and binding that causes them to remain in the system for a longer period of time. When we consider this in egg-laying species, such as seabirds, there's a major concern that the metal complexes that are associated with the lipids that are stored in either the adipose tissue, so that fat, or in the liver specifically, can be shunted into the developing egg. So as a part of egg development, won't get into all the nitty-gritty, but the fats that are found in the yolk are derived from either the maternal fat sources, so just fat that's stored around the body, or from the liver. So those are hepatic origin or liver origin lipids. Now, if we have all these metals, these contaminants that are getting shunted into the egg, that causes a host of issues for our developing chick, right? So that can include compromised shell formation if we have really weak shells. We've seen this previously with not necessarily metals, but with other organic pollutants like DDT. Accumulates in the shell, causes a lot of weakening and thinning of the eggshell. So you might have a mother bird that's roosting, she breaks her eggshell, that's now a non-viable egg. And unfortunately, you have the reproductive loss where you're not going to have a chick. You can also have things like teratogenic malformations, which can include missing features or reduced features. So reduced eyes, feet, legs, multiple legs. Those are all considered teratogenic malformations, and an overall reduction in survival rate, right? So all of these different things can kind of coalesce together to create a reduction in the number of chicks that might be viable, that'll make it to adulthood and be able to reproduce and kind of carry on for the species, which is pretty sad for our little chick here. Now, how does this translate or extrapolate to our adult birds? Well, heavy metals, like I said before, they are naturally occurring metals, and I'm grouping them together and saying heavy metals for everything. For my purposes, I'm also including some metalloid elements like selenium, but in general, they possess a high atomic weight and a density that is at least 5 times greater than that of water. Their use is pervasive in agricultural applications as well as in pharmaceutics, in tech applications, a whole bunch of different things. And the disposal of these contaminants via industrial effluents, so not just from mines, but also just in general from industrial plants that are producing so many different products that we use on a day-to-day basis, that effluent has resulted in their ubiquitous or worldwide distribution in the marine environment, but just also in the environment as a whole. Uptake of heavy metals occurs via inhalation, so if you're breathing it in, cutaneous absorption, so through the skin, or most commonly through ingestion of contaminated food or water, while heavy metal toxicity is dependent on the chemical species, so whether that is arsenic, if it's lead, copper, nickel, some of these are actually required for different physiologic mechanisms, but if you see them in high enough concentrations, it's a no-go, it's really bad. But the chemical species, so what type of metal is a huge factor, but also the dose, so the quantity that's being introduced into the animal, the route of exposure, so whether it's through the skin, inhalation, ingestion, and the physiologic parameters of the exposed individuals, so whether or not they're male, female, are they feeding young, are they healthy, are they a little skinny, are they well-fed? All of these things come into play and can affect how the organism is able to respond to that heavy metal exposure. But across the board, chronic exposure will typically cause some kind of severe physiologic detriment, which can range from endocrine and neurologic disruption to acute cell death or apoptosis, which is a really bad case. Certain metals such as copper, zinc, and manganese can exhibit partiality in their distribution once absorbed, which results in a kind of disproportionate accumulation of them in certain tissues. Like I said before, how fat likes to accumulate certain metals, that definitely comes into play here. And this doesn't just apply to bodily fat stores that might be present around the organism. It can also include really fatty tissues like the brain, which obviously has major implications when it comes to neurologic issues that might precipitate as a result of heavy metal exposure. And these are typically found in the form of lipophilic metal complexes, and they have been shown to promote absorption and tissue penetration. So they're more likely to actually be like acquired and kept within those tissues. And like I said before, they also tend to accumulate in organs that are responsible for any kind of toxin elimination. I have a little example here of a bird that came into the South Florida Wildlife Center when I was a triage nurse. It is a red-shouldered hawk who was suffering from, I believe it was lead poisoning. I think he consumed a rodent, or probably, we don't know exactly, but it was some kind of small animal that had some kind of lead pellet in it, probably had been shot by someone. Bird came in, had ingested some of that, was dealing with some of those symptoms, predominantly a lot of neurologic issues. In this image, you can see his mouth is kind of agape. He was dyspneic, so he was having a hard time breathing. He's got his wing droop, really not doing well at the time. But through just supportive care, he ended up making a full recovery and he was fine, thank goodness. But it doesn't work that way for a lot of animals, especially ones that are just out and about in the wild and don't necessarily have veterinary intervention to help them get through that tough time. So while heavy metals can be eliminated through several means, including excretion, so if they're just defecating or urinating it out, or deposition into feathers, we see that a lot across bird species. Offloading into eggs is actually a significant pathway for heavy metal elimination in female birds specifically. When embryonic exposure to heavy metal occurs, a lot of these birds are not going to be as lucky as our little red-shouldered hawk friend here. In the second image, we can actually see a comparison of normal black-necked stilt embryos and those that were deformed from exposure to selenium. So that's one of those metalloids that I had mentioned before. Now, these deformities occur because in high enough concentrations, even essential elements like selenium can act as teratogens or chemicals that can induce developmental malformations. So accordingly, there's a really significant concern for the impacts that heavy metal contamination has on chick viability. Now, all that good information, let's apply that to the Humboldt penguin. A little bit of background on them. The Humboldt penguin is a member of the order Sphenisciformes, not the easiest thing to say, but it is a group that includes all 17 species of penguins. Humboldt penguins, in particular though, belong to the genus Spheniscus, which also includes the African, Galapagos, and Magellanic penguins, who are there in order. They are a medium-sized penguin. They're about 2 feet tall and about 10 to 11 pounds, so they're kind of a smaller bird, I guess not in the grand scheme of things, but compared to some of the species I'm used to seeing, they're, I guess, a mid-sized bird. Although their size can fluctuate depending on their sex, their breeding status, and the prey availability in their environment. They live for about 15 to 20 years in captivity, or if they are under managed care, although it's kind of hard to provide a definitive lifespan for the birds in the wild because of predation and a lot of other factors. They breed twice a year, which is a huge thing that we're going to talk about in a second, and they produce about 1 to 2 eggs per clutch. If they're lucky, they'll produce 2 if you have a lot of nutrient availability, if it's a good season for them and there's a lot of prey, you're going to see birds producing more often. They'll be producing those 2 eggs per clutch. They have been categorized as vulnerable from or by the IUCN for about 2 decades due to significant population fluctuations and an overall reduction in breeding colony abundance. So like I said before, there are a lot of anthropogenic impacts at play here that are causing those population declines. As of the most recent report, there are approximately 24,000 birds globally. And when I say globally, I specifically mean in their range of Peru and Chile. So this species is found along the southwest coast of South America, from La Foca Islands in northern Peru to the Punihil Islands off of the southern coast of Chile. Their range falls within the Humboldt current system, which is a highly productive ecosystem that is characterized by elevated primary productivity, which results from coastal upwelling of cold, nutrient-dense water. So a little bit of oceanography background. Basically, it's just a physical process that happens in coastal areas that supports a lot of biomass, predominantly in the form of zooplankton, so super tiny planktonic organisms, and forage fishes that are going to predate on those zooplankton. And this then supports a massive abundance of higher trophic level predators. And when we continue scaling this up as we work through the food web, we're eventually going to get to our species like the Peruvian fur seal, the South American sea lion, and the approximately 24 species of seabirds that we see in this ecosystem. One thing that we do see in the Humboldt current system is that it is subject to a lot of wide-scale fluctuations in climate and oceanography, and that is caused by spatiotemporally cyclical ENSO, or El Nino Southern Oscillation events. Those events can take 2 different forms. It's either a warm phase, which is El Nino, or a cold phase, La Nina. Basically, El Nino means we got the depletion of prey, not so great, a lot of population declines, a lot of food scarcity, and La Nina is the opposite. So we have colder waters, great productivity, a lot of food availability. A lot of the organisms are doing a lot better during those times. So that's something that we have to take into consideration when we're discussing species that are found in this area, including the Humboldt penguin. And in the midst of all of this activity is our field study site, which is the Punta San Juan Marine Reserve, and that is the most significant Peruvian colony for the species, the Humboldt penguin. About 36% of the total population for the country, for the nation of Peru, resides at this single location. And like I said before, it is the study site for the wild birds that were surveyed in this project. Okay. So a little more natural history. These birds generally feed at really shallow depths on schools of anchovies, herrings, and silversides, as well as some other small forage fish species, but predominantly on the Peruvian anchoveta. They also opportunistically predate upon squid and crustaceans, as well as stomatopods and isopods, although those make a really small component of their diet. It's not their primary food source. And their foraging does occur over a really wide area, with birds traveling up to 35 kilometers away from the rookery on foraging trips during the chick rearing period. because of that food demand from those chicks. But during incubation periods where they're not necessarily tied down to a chick back at home that they have to make sure that they get back in time to feed, they can travel as far as 72 kilometers away during incubation when they're just nesting, they're watching an egg, but they don't necessarily have a chick to go back and take care of. So when it comes to breeding, Humboldt penguin nests take a variety of forms. Like I mentioned before, they do like to, or they solely nest in guano. They preferentially select high elevation areas where the guano deposits are sufficient for burrow excavation. They do like to dig out a little cave and nest in there, but they can also utilize surface scrapes, which are literally just what it sounds like. It's a little divot in the surface that they will lay their eggs in, as well as rock crevices and sea caves. The guano that the birds use as nesting grounds consists of the accumulated excrement of several species, which are grouped together to make our guano birds that are found in Peru. And these include the Peruvian pelican, the Peruvian booby, and the Guanay cormorant. And we see 2 of the 3 species of pelicans and the cormorants that are pictured here on the lower right. As I mentioned previously, breeding in Punta San Juan, but also just throughout Peru and Chile, peaks twice a year, around April and May, and again in August and September. Pairs are often able to produce 2 clutches per year when we have really good prey or food abundance, and more than 70% of those clutches are double broods, which means that we have 2 chicks per nest. So the 2 clutches, that just means that we have 2 breeding events. So we have 1 clutch in the spring, 1 clutch in the fall, and in 70% of the cases, we'll have 2 chicks produced at each time period. So that would be 4 per year. And this enables the breeding pairs to mitigate some of the clutch or chick loss that might be attributed to poor food availability. If things change really quickly in this environment, maybe we don't have as much fish available, one of the chicks isn't able to make it because the other one outcompetes it. Maybe there's predation. There are feral cats, dogs, and also invasive rats that are present throughout this area, but including on the marine reserve. So those guys could be predating on the chicks or the eggs before they even hatch. There are also floods depending on how the seasons are going. If it's an El Nino, La Nina, if there's a lot of rain, the nest can get flooded and the eggs can actually drown. Or you can have nest abandonment for a variety of different reasons. But producing those 2 clutches per year with 2 chicks per clutch really helps mitigate or offset a lot of these potential effects. So after hatching, the chicks require an immense amount of parental investment in the form of food provisioning, as well as observation, because they want to make sure they're fending off any potential predators, which is a lot for these birds to handle. Like I mentioned before, those 30-kilometer foraging trips to make sure that everyone back at home is well fed are a tremendous amount of demand on the parents. After fledging, the juveniles spend several months at sea foraging before returning back to their natal site. So they will return to where they hatched, and they will typically live there for the rest of their life. And we got some good images here of birds that were raised in a managed care environment. So those guys, if I'm not mistaken, are from the St. Louis Zoo. And then we also have just a great image of an adult bird that is feeding a juvenile there. They're very dependent on their parents for an extreme period of time, kind of similar to some other species, some might say. So there are multiple initiatives to protect Humboldt penguin populations that have been established, including the Punta San Juan Marine Reserve, or the Punta San Juan program, which is located at the reserve, and that is a 133-acre headland in southern Peru. It is home to the country's largest rookery, like I mentioned before, as well as a lot of other bird species, including gulls, terns, cormorants, pelicans, and the 2 pinniped species that I mentioned that I actually studied in undergrad way back when. So a huge diversity of high trophic level organisms are occupying this single marine reserve. It's a wonderful place. It offers protection to the penguins by separating their valuable nesting grounds from the surrounding Marcona district. They actually have a concrete wall that kind of subdivides the region, so they don't have to worry about as much of an anthropogenic impact as they would otherwise see if they didn't actually have that barrier there for them. And like I've said multiple times, this was the field site for the study. Got a little logo for the Punta San Juan program that is run there. The other study site was at the Brookfield Zoo and Chicago Zoological Society, although I believe they've reorganized, so it's just Brookfield Zoo Chicago now. Very exciting. But like I said, the captive or managed care component of the study was based out of this zoological facility. The penguin breeding program is part of a North American population that is genetically and demographically managed by the Humboldt Penguin Species Survival Plan. So species survival plans are pervasive. Any kind of organisms or species that are under managed care at zoological facilities are typically part of a species survival plan, where they're just making sure that the entire population is healthy, they're genetically diverse, you don't have a lot of inbreeding that could propagate a lot of different genetic conditions. Want to avoid all of that. So the Brookfield Zoo is participating in that program for North America, and they kind of will either send out or receive birds depending on whether or not they want to diversify their population genetically. So it's a really incredible program that they're running there. So the methodology to actually conduct this study, get into the meat of it. To assess heavy metal exposure in the wild population, analyses were run on archived eggshells. These shells were discarded after the chicks hatched, at which point they were collected from the nest. And the sampling took place between 2008 and 2009. A lot of these eggs actually fell into that stable isotope initial project that I mentioned. These eggs were all collected as part of a population health indice that was produced or performed at Punta San Juan by the Brookfield Zoo and the Chicago Zoological Society's veterinary teams. So they went out there, surveyed the birds, collected some eggshells, and shipped them off to us. To assess heavy metal exposure in the managed care population, serum, so taken from whole blood, you have different components, but the liquid component that is acellular, it's not comprised of either red blood cells or white blood cells. That serum component, as well as eggs, were collected from the birds at the Brookfield Zoo and then sent to NSU. The serum essentially just provided a baseline for the circulating metal concentrations during the non-breeding period. We collected it during a time where there was no chick rearing, no eggs, nothing going on. And those samples were actually collected well after the last eggs were laid to kind of just act as a control and see how much metal was floating around normally. The eggs were removed from parents and stored in plastic specimen containers under refrigeration before they got shipped down here. Same with the whole blood, which was actually frozen and then shipped down here. And that is how we got our samples. It is key to note that the eggs that were removed were no longer destined to kind of produce chicks or adult birds eventually that had good genetic representation within their population. The parents had bred multiple times. You would have had too much genetic expression or representation of that particular pair together. So they would be removing these eggs and discarding them anyway. So instead of just disposing of them, they sent them to us. And they actually have a program, because this is a long-standing thing that they've been doing, where they have dummy eggs. So they go ahead and replace the ones that they're taking with the dummy eggs so that the birds can still go through the rest of the reproductive cycle naturally. And then when they get to a time when the egg would be hatched, then they will remove it and kind of allow the birds to proceed from there. So since each egg component has a different function and is comprised of different materials, whole eggs from birds under managed care were separated out into eggshell, albumin, and yolk. And I have the process shown here, where I kind of gently crack the egg along its midline. I separated out the egg yolk from the albumin, which you're probably familiar with if you've done any kind of baking. You got to separate the egg from the yolk if you're doing anything high tech. And I went ahead and placed those respective components into a Teflon digestion tube. Now, before I cracked the eggs and separated out the contents, I did go ahead and clean them using some, deionized water, scrubbed them, made sure everything was all good to go. I wanted to make sure that I had removed any guano for potential contamination, and same for the eggs that came from our archives from Punta San Juan. They were just cleaned using the same technique. So prior to digestion, all the samples were dried, went ahead and digested them using acid solution. We use a conventional microwave oven, and these amazing little, they're like digestion blocks. They're incredible. You put your sample in them, you throw it in the microwave, and it like ultra pressurizes them so you can actually break down organic material incredibly quickly. So we did that, got everything processed, sent out to the University of Southern Mississippi so that we could test for trace element analysis using ICP-MS, which is just inductively coupled plasma mass spectrometry, a very long name, incredible technique. And we looked for 16 metals in total, which were aluminum, arsenic, cadmium, cobalt, chromium, copper, iron, mercury, molybdenum, nickel, lead, selenium, a metalloid, not a metal technically, tin, vanadium, and zinc. So very long, but I just wanted to get those all out here. And this is the data that we got back. A lot of numbers. Key takeaways from this right off the bat are that all of the egg components, so we had 72 egg components in total when we parsed it out, and we have all of our different albumin, yolk, eggshell, everything contained detectable values of 13 of the 16 elements, which is incredible. Arsenic and molybdenum could not be detected in the managed care penguin eggshells, probably because they didn't really have a lot of environmental exposure. And we also couldn't find mercury in the wild eggshells, so that is good news. For the metals that were below the detection limit, we went ahead and proxied them. We did some cool stats behind the scenes to make sure that we were estimating potential exposure risk, just so that we weren't completely dismissing those data points. We want to say half of our potential detection limit, so a very small value, was kind of factored in. Like I said, nitty-gritty stat stuff, but just to make sure that we're actually taking into account any potential metal exposure that these birds might have been dealing with. So all the 16 metals were detected in penguin serum, but those values were way below those identified in the egg components for either the wild or the managed care individuals. This disparity is probably explained by the turnover rates for these different tissues. For serum, it provides a snapshot of the current circulating heavy metal loads throughout the body, whereas the egg components are kind of accumulating and retaining the contaminants throughout the process of that egg formation, so they can just store more metals, which is why it seems like their concentrations are so much higher. And again, that kind of has major implications when it comes to our chick, right? Because you're accumulating all these metals. How's that going to affect the developing chick? So I wanted to show you a quick graphical representation of this data and subdivide it out by our different sample types. What we see here is that wild eggshell had the highest concentrations of aluminum, cadmium, and vanadium. Pretty interesting. What does this really mean? We have this lovely scanning electron microscope image of a cross-section of an eggshell off here to the right, because there's some pretty interesting physiologic mechanisms that could be producing the results that we're seeing here, specifically when it comes to cadmium. So we normally have calcium that is present within the eggshell. The thing is, both cadmium and lead are very similar when it comes to their chemical species status, and they are capable of mimicking and replacing calcium during shell biomineralization. The replacement of this calcium with its mimics results in the inhibition of calcium precipitation into those tissues. So we got no calcium, but we do have the inclusion of both cadmium and lead. Subsequently, we have those heavy metals that are concentrating in the shells in lieu of calcium, which also has a lot of different effects when it comes to structural integrity for the eggshells. Not a good thing. An unusual, very unexpected result was that nickel was highest in the managed care eggshell. It is an essential metal, so it's important that they have it to some extent. But I had hypothesized that this would concentrate to a much greater extent in the yolk, because obviously the yolk is going to be used as a nutrient source for our chicks. However, found a lot of it in the eggshell. I did perform some preliminary analyses on prey items from both Peru and the Brookfield Zoo, and it seemed like the prey items that were provided to the birds at the zoo were just extremely enriched in nickel, compared to the prey that was being consumed by those wild individuals. So that's probably why we saw that, but it's something interesting, notable. So highest concentrations of aluminum, there we go, sorry, copper, mercury, and selenium were observed in albumin, which is a lipid-free fluid. It's kind of just what everything is floating around in as the chick is developing. And we can explain the presence of these metals by some of the proteins that are comprising albumin, which include ovotransferrin and ovalbumin. Those are the two biggest players here. And they form metal complexes that offer protection from bacterial infections, which is super important. If we can assimilate metals that are normally part of just this aqueous material that we're floating around in, and use that to ward off any kind of bacteria that could be invading and potentially threaten the chick, that's phenomenal. The only issue is that it also causes potentially excessive metal accumulation within the albumin, which can then have adverse effects. Ovotransferrin here on the left is specifically important when it comes to iron binding, but it plays a role in a lot of different metals, including aluminum, cadmium, chromium, cobalt, kind of a lot, the whole gamut there. So, well, sneak peek of what I was going to talk about, but I'll just go ahead and do both of these now. The range of metals that were found to concentrate in yolk, vast. We can largely explain these by the metabolism that is responsible for avian vitellogenesis, or yolk production. That's all that word means. Lipid is the largest yolk constituent or component by dry mass, and the lipids that comprise that yolk, called yolk-targeted very low-density lipoproteins, or VLDLY, and vitellogenins, are synthesized in the liver. So if we go back to that image that I had produced a while back, where it's like the sunset and we were talking about the organs that come into play, how we get those fats that are derived from both the adipose stores that are present within the mom and also the liver, this is what I'm talking about. So these fats are synthesized into the liver, or they're synthesized in the liver, and then they are secreted into the circulatory system. So they're going out into the blood, they're taken up in by the ovaries follicles for the generation of yolk when the bird is forming an egg. And this yolk is then the nutrient source for that developing chick. So when we have these metal complexes, they're bound to these lipids that are in the liver. The liver is already responsible for trying to shunt all these metals out of the body because it's like, your toxins, I'm trying to eliminate you, get you out of the system. Well, it's interacting, those metals are interacting with the lipids. They're retained in the liver to some extent, unfortunately, same as they are also retained in the adipose tissue. Those eventually get liberated as the mother bird is attempting to generate an egg. And those kind of get offloaded from the mom into those egg products. Some previous research has shown that in adult female birds, in the plasma, vitellogenin concentrations increase, and that corresponds to a rise in trace mineral concentrations because those vitellogenins are binding to the metals that are present in the system. So because these different lipoproteins, those vitellogenins and the VLDLYs, exhibit a really high binding affinity for a wide range of metals, particularly iron and zinc, that's probably why we're seeing the majority of the metals that we tested for concentrating in the yolk, which again, really not good because that is going to be our nutrient source for the developing chick. So it's getting that immediate exposure straight off the bat, a lot of metals. So what are the main takeaways from these results, right? A lot of data. The values that were measured in the managed care and wild eggs do not meet thresholds for toxic effects in adult birds, which is good news. But do they meet toxic thresholds for chicks? Not very well established in the literature. They are similar in value to concentrations that are detected in the eggs and tissues of wild seabird species, including other penguins, which is incredibly surprising for our managed care individuals. We're expecting them to not really have a lot of metal exposure because they are in what we would consider to be a safe environment, right? They're not out there dealing with a mine efflux. They're not dealing with just general anthropogenic impacts. They're kind of safe and isolated within a zoo environment. So we're not really expecting that, but we're still seeing those same numbers. This is really important because it demonstrates that even though the managed care birds are living what you could consider to be a low risk lifestyle, as much, as a bird could live, they're still concentrating metals in their eggs to the same extent as the wild birds. And a recent publication from 2022 noted that Punta San Juan in particular had the worst environmental conditions out of any of the Peruvian marine protected areas due to the heavy metal contamination coming from those local mines. So if we already know that our managed care birds with very limited exposure to contaminants, except for what's being provided through like a presumably safe, diet, are already concentrating metals to such a great extent in their eggs, we can only imagine how bad it's going to be for our wild individuals. Additionally, the wild eggshells that I used in this study were collected about 15 years ago now, and they may not accurately reflect our modern populations, what they're going through as far as their metal exposure. So as such, the potential for our heavy metal exposure during embryonic development and subsequent reductions in offspring viability are a major concern. And this continues to be like a multi-decadal population decline causative agent for that. So where to next, right? Well, at the end of my master's, I got awarded a grant. It was the Wild Animal Health Fund that was provided by the American Association of Zoo Veterinarians. I kind of submitted this project or an extension of this project to them, and they were like, this seems like good work, and we care about this species. So they funded that project. At the same time, I had also applied for an in-house grant at NOVA, which was the presidential or PFRDG, President's Faculty Research and Development Grant, which we were also awarded. I could not find any good images on their website for any kind of, title or anything. So I did put a little picture of Dr. Hirons because they awarded it, and they put her picture up there. And they actually said that this was the highest ranking protocol received for that year. So this time last year. So very exciting news. And that sounds like a PhD to me, which is how I ended up here as a doctoral student continuing on with the study. So the goal of my dissertation is to provide a comprehensive assessment of heavy metal offloading pathways through the analysis of prey, so the contaminant source, and penguin blood, egg components, feathers, and excreta from birds that are under managed care at the Brookfield Zoo. Since it is not advisable to collect whole eggs from declining wild populations, obviously we don't want to take any potential, new birds away from those wild populations, the managed care birds are going to allow us to generate a statistical model, which can then be used to interpolate metal concentrations when we might not be able to acquire a full sample set, such as a whole egg. So I have this figure here. I feel like it's probably way more efficient than I can ever be when it comes to explaining what the goal of the project is. But essentially, we are looking at everything possible from the birds, whether it's their food, their feathers, their blood, anything that we can take, we're going to take, assess for heavy metals, and see if we can put together a large picture showing where all those heavy metals are going. So from when they enter into the bird's body as a fish to where they're excreted, either through feather production, through feces, if it's in the egg components, getting all of that quantified, putting a number to it so that we can kind of create a comprehensive image and creating relationships, numerical relationships that are essentially a model and we can use to backtrack. So if we have, say, an eggshell, can we use that to interpret what the mother's serum concentrations for arsenic were at the time of egg formation? Through this captive study, we'll be able to do that because we have an incredible population of birds that are extremely consistent. They are always eating the same food. We don't have to worry about environmental perturbations. We don't have to worry about ENSO effects or whether or not this mine is just discharging more efflux than normal. Everything is incredibly consistent, so it provides an excellent opportunity for us to be able to study this process in this species. So methodology, not going to run through this like crazy. Essentially the same thing that I just talked about. We're going to be getting blood, eggs, excreta, also feathers from these managed care birds. We're also going to be sampling the prey. Got them here. And we're going to use that to generate our model. And a whole lot of mumbo jumbo. Basically, they're called linear mixed effect and fixed effect models, and those are going to assess the relationships between the metal concentrations across all of our different tissue types. These are going to interpolate those egg components, and interpolate is basically just a fancy word for infer what those egg concentrations are going to be. If we can't necessarily get egg yolk, but we can get the shell, we can do the math and figure out what those yolk concentrations probably would have been in a wild population if we were able to actually collect them in the first place. And this will allow us to reliably predict the embryonic exposure levels and thus the toxicity risk that chicks might be undergoing, right? So if we can figure out all the math and figure out how much arsenic is accumulating in the yolk of an egg through just collecting the eggshell so that we don't have to disturb a nest that has active chicks being reared, or we don't have to collect a whole egg and potentially remove that bird from contributing to the population later on, that's phenomenal. It allows us to really add to the health indices that are being conducted on this extremely vulnerable population. So we have the statistical models, which are then going to be shifted over to our wild population. And like I said, we're going to be collecting things that are a lot less invasive. So if those are discarded eggshells, if it's feather, excreta, all of those will be collected in addition to blood samples. Those blood samples are normally just collected from these birds to make sure that they're healthy as a part of their routine veterinary checks. And we're going to put all this data together to make a comprehensive image of what is currently happening for this wild population so that we can better inform management and conservation efforts that are being initiated and continued at the study site so that these birds can be conserved long term. And I'm very excited because the upcoming field season actually begins on April 29th. So on the 29th of this month, we will be going to Peru and sampling these birds and collecting a whole lot of good data. And we'll be there for about a week and a half. So very exciting. And I cannot wait to come back and inform my NSU community about what we found. So gonna call it here, see if there are any questions.
Awesome. Well, I want to start by just thanking you for letting us listen into that. That was absolutely fascinating to listen to. And I did see some questions that did come in. So one of them is kind of starting back to the something you mentioned at the beginning. You mentioned you were able to get published. I know this is a little bit different from the topic itself, but how was that process? I know we do get published here as a research university, so some people were just curious how that process went for you.
Yeah. So the publication process was really an interesting experience for me as a graduate student, because obviously that's everyone's goal, right? You hear this throughout your time from when you are an undergraduate first looking to become a scientist. It's publish or perish. How is that going to actually pan out for me? How difficult is that process going to be? Well, after I completed my master's thesis, I had this wealth of information that was contained within my thesis, trimmed it down into a manuscript, way less content. And it's a little heartbreaking to be like, I've spent so much time finding all this incredible information. I need to condense it down into this 6-page paper. How will people appreciate how much work I put into this? Don't worry, they will. You submit that off to a journal. Generally, you have 2 to 3 reviewers from various organizations or entities, whether it's a university, a research group, that are going to be reviewing your manuscript for its scientific validity, for the content of the paper. They will go over like writing, if they prefer a certain technique or a certain style. They will talk to you about that. But essentially, their main focus is making sure that the work that you're relaying to the scientific community is actually valid, that the study design was correct, and that you have good information that is sound, that you're trying to relay to the general public and, benefit the scientific community at large. So that was extremely rewarding. I got a lot of incredible feedback that gave me a lot of insight and a lot of ideas for where I kind of wanted to adjust things and tweak them to make them better and explain things more fully. So that was a great process. Got that back. Some edits, nitty-gritty stuff, making sure that everything is grammatically correct, formatted to the journals, but overall an incredible process. Very rewarding to have someone from outside of your institution validate that your work is actually something that is incredibly worthwhile, because a lot of us are operating in our little bubble within a university, and it's nice to have people from outside that space be like, hey, you're doing good work and we want to publish this. And when you finally get that email saying, hello, your proof is completely good to go, you'll see this on the website in, a couple weeks, it's extremely rewarding. Definitely a good process.
Awesome. That's great to hear. Definitely getting that external validation's gotta feel fantastic on top of what NSU students already do. So actually, the next question does kind of tie into that a little bit as well. So do you mind talking about how much flexibility are you allowed during your research at NSU? Do you work well with the professors? Like, how much communication does it seem easy to work with our faculty members?
Definitely, for sure. I have a very diverse committee of NSU faculty as well as some external individuals, mostly out of the Brookfield Zoo, so some veterinarians. But yeah, the faculty at Nova have been incredibly supportive. It has been a very fulfilling and rewarding time. I also say that with the, I guess, perspective of having done my undergraduate education here too. So it's very interesting seeing the progression, seeing your own progression sometimes through your faculty advisor's eyes. So you kind of perceive how they see you when you first come into the program. As a new graduate student, you're kind of like, I don't know what I'm doing. I just want to study sharks or corals or birds or whatever. And seeing how you're able to progress through that, you can kind of pick it up when you're interacting with them. And it's very rewarding to see when they kind of are proud of you and they're like, you did a good job. So I would say that the NSU faculty are incredibly supportive. They have been very helpful, especially when it comes to things that I was way out of my depth on, like statistics, not my strongest suit. So people have been incredibly helpful. And even folks who were not on my committee were always more than happy to lend an ear or give me some advice for how I could, mitigate or, deal with some issues that I might have been facing along that project.
Awesome, thank you. Next question is kind of there's a couple that actually kind of work well together, so we're gonna it might be a little bit of a reading. First question was, what is something that you can see from the future down the road from a global impact, and then kind of tagging into that, have you been able to work with any policy stuff like in Peru specifically? Like, have you been able to, connect? Like, are there any future plans specifically with Peru that you might be able to help out with, and then kind of global impact, kind of looking down the road for you?
Yeah, so as far as global impact stuff goes, I mean, that's the dream for everyone who's working in conservation, right? We all want to make sure that there are best practices in place whenever it comes to anything that is an anthropogenic impact. So if that's, mines, making sure that they're disposing of their waste properly, or maybe changing the strategies that they're using and being mindful of the fact that they are so close to a coastal environment. I think that these data will definitely help validate that this is a concern. So previous research at the same site has already proven that the wild population was exposed to heavy metals to a pretty great extent, especially for things that were found in mine efflux. So that already off the bat kind of substantiates that it is a concern. And I think that this project just goes to show that it's not just the adult birds who are being directly impacted because, we see birds are caught in oil spills. We can kind of envision that the adult animal is dealing with these consequences. But a lot of times people might not realize that it's also those subsequent generations that are already being impacted before they even have the chance to waddle out into the ocean for the first time. They're already having to deal with some of those effects. So globally, I mean, I think this just contributes to the wealth of knowledge that a lot of our human activities are impacting the environment and the animals, which is already well known. It's just kind of nice to have, I guess, additional data points to really show that this is a major concern. When it comes to conservation strategies and management that is currently ongoing in Peru, I'm very excited to be going down into the field for the first time in a couple of weeks. So that's really nice. I'd love to continue collaborating with them. And as you actually mentioned during my introduction, I am pre-veterinary, which is crazy to say that I'm still pre-something as a doctoral student, but the goal is eventually to get into veterinary school. And one of my committee members has been incredible in kind of guiding me along the path of pursuing a career as a wildlife veterinarian. So I'd love to do work really long-term in conservation by, participating in some kind of management consortium as a veterinarian and providing that on the ground health and disease, we're collecting samples kind of work. So I think that this is a good setup for that kind of career long-term. I hope.
Absolutely, and we hope so as well. You did touch on this a little bit, obviously, kind of toward the end of your presentation. The question came in is, you're having the ability to travel. Obviously, you have that trip coming up, which obviously I didn't know about. That's really exciting. How much more travel do you anticipate, kind of, getting to do with your research?
So I am anticipating to at least have one other field season in Chicago, so up at the Brookfield Zoo. We're in a lot of talks about getting that off the ground and having me go up there. It'll probably be near the end of this year. So it'll be for their fall breeding season. It does kind of fall after the breeding season for the wild birds, so it'll probably be around November. But I've got the upcoming Peru trip, definitely going to the Brookfield Zoo at some point. And there are some discussions about a potential second field season in Peru because, like I said, the birds do breed twice a year. So that might also be, something to look forward to. I'm really hoping so. And outside of that, definitely participating at conferences, traveling for that. I've had a lot of great experiences locally at conferences, one of which my primary advisor held. So just being able to present, meet people who are working in the same fields, network, a lot of opportunities for that coming up, especially as we start getting this data in and I start being able to submit abstracts for conferences or submit talks as we kind of get that data in and I can actually show what is currently happening in the field currently. So looking forward to some more field seasons as well as some travel for some conferences as well.
Awesome. And I want to make sure I'm, cognizant of your time, Maya, as well, because it sounds like you are crazy busy. So I do have just one final question. It's actually going to come from me, because I don't think I would be doing you a disservice if I didn't ask at least. But, obviously I know a little more about your story. I know you've been at NSU for a very long time, which is so amazing just hearing your story and everything about you. But, why did you pick NSU? What really drew you to our university?
That is a very interesting question. I think I might have more of a unique backstory, not to, inflate my ego or anything, but
Brag away, brag away.
No, no. I think I have a bit of an odd story for how I came to NSU. I was a very young applicant. I graduated from high school when I was 12. So my parents were not really willing to let me go out of state or really anywhere further than about a 30-minute drive from my house. So I came to Nova kind of with just the plan of I want to be a veterinarian one day, and that's about it. But I was a very young applicant, and from the get-go, NSU was phenomenal. Admissions was incredibly nice, especially looking back. It's a little odd having a 12-year-old come in, a little bespeckled self, and being like, hello, I'm here to attend Nova. I'm a college student. But yeah, like I said, from the very beginning of my studies here at Nova, admissions, incredible. All the faculty, all the mentors that I've had through my undergraduate education were absolutely phenomenal. Everyone was incredibly understanding, supportive. The NSU community, whether that was clubs, professional development, research was obviously a huge portion of my undergraduate education, but all those things came together to really make for a phenomenal college experience that is, again, looking back, incredible how comprehensive it was since I started university at such a young age, how supportive Nova was throughout that entire process. And then when I graduated, 16, a little too young to go to vet school still. And I was like, you know what? I've had this incredible opportunity to do research. I'd really like to carry that forth. Dr. Hirons, again, talking about faculty who are supportive. She's amazing, was like, absolutely, more than welcome. Please come in as a master's student. Began my master's, kind of got things delayed a little bit with COVID, but still everyone was incredibly helpful and supportive throughout that entire time. And like I said, at the end of my master's, I got a good amount of funding and I was like, this is an incredible project that I have really dedicated a good portion of my life to. Now it's almost half my life I've been at Nova, and doing research here. And I was like, I cannot miss out on this opportunity to do incredible work with incredible people on an incredible species. So I was like, I think I'll stick around here. And that is how I am now a doctoral student. But truly a phenomenal experience. And I could not be more appreciative of how supportive and amazing Nova has been throughout this entire process of personal and professional growth. So very appreciative.
Well, that's great to hear, Maya. Obviously, I know, from my staff alone, we're very excited to see where you go next. I know you got, plenty of time to finish up here with your PhD and everything. I know you just got started. But we obviously couldn't be more excited to see where you go, and, just so happy that you've been able to stay at NSU and clearly have such a good experience. I know personally Dr. Hirons very well. I see her every day pretty much at the office, and she gushes about you whenever she gets the opportunity to. She probably thinks of you as another child, I'm sure, at this point. So that's great to hear, Maya. So, for those of you that are watching later, that were with us this evening, thank you so much for being here. And hopefully we're looking forward to calling you, Dr. Maya soon, of course, or Dr. Daniels, or however you would like to be pronounced her name. We're very excited and thankful very much for you to be here with us this evening. Thank you so much. And Maya, thank you once again. And we look forward to, seeing you around campus and talking to you again soon. I hope you have a great rest of your night and good luck with all of your research. Take care, everybody.
Bye.